Welcome once again to Population Genetics and Evolution Bio 208, Unit 2, Session 4, Analysis of Polygenic Inheritance. And by the close of this session, you should be able to state the components of polygenic variation and explain, and also analyze polygenic and interpret polygenic data. Polygenes are genes with effects that are too small to be individually distinguished. They are sometimes called minor genes. In polygenic inheritance, segregation occurs at a large number of loci, affecting varied genetic traits referred to as phenotypes. When a man with a homozygous condition for dark skin marries a woman who is homozygous for light skin color, their progeny in the first filial generation will have an intermediate color, mulatto, in the skin different from that of both parents. The first filial hybrids form 8 different types of gametes in each sex, giving rise to 64 combinations in the progenies of the second filial generation. The skin color of the individuals in the second filial generation varies according to the number of genes which they inherit for skin pigmentation. The skin color ranges from pure black, that's dominant A, dominant B, dominant C in 1/64 individuals, very dark brown in 6/64 individuals, dark brown in 15/64 individuals, mulatto intermediate in 20/64 individuals, light brown in 15/64 individuals, very light brown in 6/64 individuals, and pure white in 1/64 individuals. The phenotypic expression of polygenic traits is subject to significant modification by the variation in environmental factors to which organisms are subjected. Polygenic variation is made up of 3 components: phenotypic, genotypic, and environmental components. It is the total variability which is observable and determined by both genotypic and environmental variation, and hence changes under different environmental conditions. Such variation is measured in terms of phenotypic variance. Genotypic variation, it is the inherent genetic variability which remains unaltered by environmental conditions. This type of variability is more useful to a plant or animal breeder for exploitation in selection or hybridization. Such variation is measured in terms of genotypic variance. The genotypic variance consists of additive, dominance, and epistatic components. Environmental variability refers to non-heritable variation, which is entirely due to environmental effects and varies under different environmental conditions. This is uncontrolled variation, is measured in terms of error mean variance. Analysis of polygenic traits or quantitative trait requires the study of large numbers of progeny from a population of organisms. Environmental variability, the number of genes may be calculated if the proportion of second filial individuals expressing either of the 2 most extreme as parental phenotypes can be determined according to the formula 1/4n equals to the proportion of offspring either red or white. 1 = 1 solve for n, that's 43 = 64, n = 3, 4n = 64, or determining the number of genes minus the bracket open 2n + 1 bracket close rule. If n equals the number of gene pairs, then 2n + 1 in bracket will determine the total number of categories of phenotypes. So in our example where there were 7 phenotypic classes, 2n + 1 into bracket equals 7 into bracket 7 minus 1 equals 6 equals 3, n equals 3 to 2. Polygenic variation cannot be classified into discrete groups, that is, variation is continuous. This is because of the large number of segregating loci, each with effects so small that it is not possible to identify individual gene effects in the segregating population or to meaningfully describe individual genotypes. Instead, biometrics is used to describe the population in terms of means and variances. The relevant statistics to be discussed are the mean, median, range variance, standard deviation, and standard error. That's genes and special statistics. Importance of population genetics. Construct statistical models based on population or quantitative genetics theory to improve prediction and selection during breeding, thus facilitate selection of parents and parental combinations in breeding. Understand natural genetic structure, which is important for determining the appropriate sample size necessary for various procedures. Applied to genetic models to identify genes that can be used for marker-assisted selection, and understand how pest populations evolve as insect weeds pathogens and thereby develop better control measures. So in review, analysis of polygenic inheritance data using the relevant statistical tools will generate the relevant information to draw scientific conclusions. Interactions of genotypes and the environment influence phenotype, giving rise to polygenic variations in a population of organisms. Polygenic inheritances generate quantitative data which can be subjected to statistical analysis, including the mean, the median, the range, the variance, standard deviation, and standard error.